A sound's frequency is the number of waves that pass a certain point at a given time. A high-pitched noise is the result of shorter waves moving in and out more quickly, while fewer, slower fluctuations result in a lower pitch. How loud a sound registers depends on the wave's amplitude, or the difference between the high and low pressures created in the air by that sound wave. Your ear is divided into three major areas: the external, middle, and inner ear. The external and middle ear are only involved with hearing, while the complex hidden inner ear is key to both hearing and maintaining your equilibrium. It's made up of elastic cartilage covered in skin, and its main function is to catch sound waves and pass them along deeper into the ear. Once a sound is caught, it's funneled down into the external acoustic meatus, or auditory canal, and toward your middle and inner ear. Sound waves traveling down the auditory canal eventually collide with the tympanic membrane, which you probably know as the eardrum. The sweet sound waves of your favorite jam collide with the eardrum. They push it back and forth, making it vibrate, so it can pass those vibrations along to the tiny bones in the middle ear. Now, the middle ear, also called the tympanic cavity, is the relay station between the outer and inner ear. Its main job is to amplify those sound waves so that they're stronger when they enter the inner ear. Parts of the membrane vibrate depending on the pitch of the sound coming through, so the part of the membrane with the short fibers vibrates in response to high-frequency pressure, and the areas with the longer fibers resonate with lower-frequency waves.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
We will now show you what the process of such an inflammatory response is. In our immune system, many cells play a part, among which white blood cells. An important white blood cell is the macrophage. The macrophage recognizes the bacterium as a non-self substance and engulfs it. The bacterium is slowly destroyed in the macrophage until only very small pieces are left. These little pieces of bacterium are made visible on the outside by the macrophage. This way, the other cells of the immune system are able to notice that a bacterium has entered the body. Apart from that, something else is happening. Since the macrophage engulfs the bacterium, it creates inflammatory substances. These substances are called cytokines. Cytokines bind themselves to receptors on cells. Cytokines exactly fit the shape of such a receptor, like a key in a keyhole. In this manner, the cytokine communicates to another cell. For example, a cytokine is able to warn a macrophage, from the position of another macrophage, that bacteria are near that have to be destroyed. This is an important step in the inflammatory response. At the point of the inflammation, the amount of macrophages then increases in order to destroy the bacteria. The macrophages are, however, not able to destroy all bacteria. The macrophages, therefore, go to the lymph nodes in our body in order to warn other cells of the immune system that an inflammatory response is taking place. These lymph nodes are important with regard to the defense of the body. Lymph nodes are information centers where cells of the immune system meet in order to exchange information. The macrophages that have engulfed bacteria at the point of the inflammation come into contact with T cells in the lymph nodes. B cells are also present there. Both are white blood cells that play an important part in the inflammatory process. T cells recognize the bacterium pieces on the outside of the macrophage. The T cells bind themselves to those pieces of bacterium on the macrophage, and they become active. The B cells also become active. After this activation, the T cells and B cells divide until a large quantity of these cells has come into being. The T cells and B cells go to the spot of the inflammation. At the spot of the inflammation, the T cells help the other inflammatory cells to attack the bacteria as effectively as possible. This way, they help the macrophages to make more cytokines, which are the inflammatory substances.